Welcome back. So in that intro video, we went through a lot of the PowerPoint. It went through the different, the DNA that different organisms have, what are their different structures between eukaryotic and prokaryotic cells, DNA replication, how can we make new DNA, how can we get DNA all the way to protein, kind of the whole central dogma, transcription, translation, and then even some of the mutations that can happen along the way when we're making new DNA. This last part is just ways that organisms can, going wrong way, ways that organisms can get their DNA from one organism, especially if they're single-celled, to another organism. So it's transferring that genetic material. Now, normally during transferring of genetic material, especially with eukaryotic organisms, there is some genetic recombination that happens. And just like the name implies, it's recombining the DNA. When you've got two chromosomes, and like us, we've got 23 pairs of chromosomes, when those chromosomes pair up, they're really close to each other, and sometimes some of that information can get kind of jumbled around. The DNA can get jumbled around. Still contain all the necessary information, it just gets mixed around a little bit from one chromosome to the next, to the paired up chromosome. So that usually happens only in homologous chromosomes, those perfectly paired chromosomes, the fact that you've got 23 pairs. And then that information now has to go in a mic from parent to offspring. It's called vertical gene transfer. And a mic, it's going top down. So organisms replicate their genomes, they make new DNA, and then they provide copies to their descendants, so the parent to offspring. That's the top way that eukaryotic organisms transfer genetic material, is that they give their genetic material to their offspring. However, bacteria, prokaryotic cells, don't get together and have babies together. They're constantly just making new DNA and passing it on. But there's some other ways that they can get new DNA as well. So they do genetic transfer. It's called horizontal gene transfer. And this is the most common method of genetic exchange in the microbes, such as bacteria, that we talk about, is that a donor cell contributes part of its DNA to a recipient cell. So one organism, one bacteria cell, is going to give part of its DNA to a recipient cell. And it can do that in one of four ways. So it's not an offspring, it's just like another adult to adult. How one adult DNA or one adult bacteria can give DNA to another adult bacteria. And so these are my next four slides, the four types of horizontal gene transfer that bacteria and prokaryotic cells can do. The first one's called transformation. So not all cells can do this. It's pretty rare, but some bacteria are more prone to it than others. And it's that the DNA can actually leave one cell. It can go through the cell wall, it can go through the cell membrane, and go from one bacteria cell to another one. Now, one of the bacteria that can do this, and Frederick Griffin was the one that discovered this in the early 1900s, was Streptococcus pneumoniae. Now, there are different strands of Streptococcus pneumoniae. One of them is called a smooth strand, one of them is called a rough strand. Now, the smooth strand is called smooth because it has a capsule around it, which, again, anytime a bacteria has a capsule, it's always good for it, not for us. And it's just smooth around the S. So we know a capsulated bacteria is a nice, healthy, strong bacteria, which means it's bad for us. So if you put this bacteria in a mouse dies. We also know that if we can take that heat-treated smooth strand, and so we call it strand S for smooth, and we kill the bacteria. If we inject a dead bacteria into mouse is going to live. But we also know that there are some strains of Streptococcus pneumoniae that can't make a capsule. So we call them rough or R strands because they just don't have that nice smooth capsule around the outside. Well, the capsule's needed for the bacteria to cause us disease. So if we take one of these rough strains and put it in a mouse, because it doesn't have a capsule, it can't hide from the mouse's immune system, mouse is going to live. Now, we know we've got different strains. They're all Streptococcus pneumoniae, but we know we have different strains. So there is different DNA between these two. So the question is, what if we mixed these two together, put them in a vial, and then injected that into the mouse? So it's kind of one of those, all right, a dead S strand, mouse lives. A live R strand, mouse still lives. So you'd expect that if we mix these two together, put them in a needle and put it in the mouse, mouse would live. But it doesn't. So Griffith's experiment, he mixed these two together, injected into the mouse, mouse died. Well, what happens when he mixed these two together, even though this is a dead bacteria, it still has DNA. There's still DNA present, dead or not. And some of that DNA was able to leave those cells, go into the living R strand, and some of the DNA that left this cell to go into the living cell contains the DNA on how to make a capsule. And so as long as the DNA on how to make a capsule goes from the dead strand to a living R strand, this now living R strand that never knew how to make a capsule, it now has the DNA on how to make a capsule, and it makes a capsule. And so that's why it ultimately killed the mouse, is that DNA went from one cell to another. It was able to leave the cell wall, the cell membrane of this cell, and enter through the cell wall and cell membrane of this cell. And and it now made a capsule. So this is just showing the DNA was broken down and it went into other strands. They acquired the ability to synthesize capsules. Now again, transformation's pretty rare. The DNA has to be able to go through cell walls and cell membranes out of one and then into another. So it doesn't happen all that often, but some bacteria are more likely for transformation. Most Streptococcus are more likely, some Neisseria. And so it's rare, but it can happen. Another way we can get genetic information from one bacteria to another, it's called transduction, is we're going to use a virus to carry it. So we're going to use a virus. So here's one bacteria cell, and we want to get some of this purple DNA into another cell. So this bacteria is going to get infected with a virus. Virus is just going to infect bacteria. And it injects its viral DNA into this bacteria. It's going to hijack this bacteria, and that bacteria is going to start to make viruses. Well, some of the bacterial DNA can get into a virus. And when this cell bursts forth and all the viruses leave, some might actually be carrying some bacterial DNA with them. So when that virus goes to infect another bacteria, some of the original bacteria DNA is now going to go into a new bacteria. So we're using viruses to carry DNA from one organism to another. Again, it's not super rare, but the fact that we know we can do this, we've actually started to utilize this, and we're starting to manipulate it into labs, that we can know that we can get some DNA from one cell to another using viruses, that we're now trying to kill bacteria using phage viruses. A third way we can get DNA from one cell to another, and we've talked about it before, is bacterial conjugation, because this is the most common way that bacteria send information from one cell to another. So this particular bacteria, it's got the genetics, it can make a pilus, and it's going to make a pilus or a pilus, different books, different pronunciations, and connect to another one. They don't even have to be the same bacterial species. These aren't. And and some of the DNA from this little plasmid, which is just a snippet of DNA that the bacteria doesn't need, but it could be useful information, some of this DNA is going to leave and go through that conjugation pili and into this cell. So we can see that it's going to double its information and send a copy over, and then it will make the complementary copy. And so most common way that bacteria send information from one cell to the other. The last way that information can go from one cell to another cell, and is they can take snippets out. And most of the time, this is times we're just taking DNA, moving it from one location to another in the same molecule, but also taking some of that DNA and moving it to another, usually by bacterial conjugation. And but we can take information, we can double it, and then we can send it elsewhere. So this is an example, it's a segment of DNA that's going to move from one location to another. So this snippet of DNA is going to move from here to over there. And this one can replicate and then move into multiple areas of the DNA segment. Again, it's one big, long, continuous circular chromosome. And but some of these little segments of DNA can go onto those plasmids, and then based on here, they can actually send some of that information to another cell. Now, anytime we have snippets of DNA, we move it from one place to another or duplicate it and send it other places. All of the segments are always going to be palindromic sequences. So again, this is DNA, so we're talking sequences of A's, T's, C's, and G's. And a palindrome means it's a sequence that's read the same way forwards as backwards. And so radar's read the same way forwards, backward, race car, mom, dad, things like that. And so we would take a sequence like ATTA. I'm like, that's a sequence that you could read the same forwards and backwards. And I'm like, and we can move that into another area. Now, a problem happens when we start moving DNA from one section to another, especially if we're replicating it and moving from one place to another, is it can result in frame shift insertions. That the DNA is not going to be read correctly when we're trying to make correct proteins. So it can happen that we can get some mutations out of it, but it is one way we can move DNA around inside the cell, but we can also move DNA onto plasmids and out into another cell as well. So that's the last way we can move genetic information around. Now you should, between the two videos, have all the information needed to be able to complete the worksheet that you have due.